Hello everyone, and welcome back to AP Bio with AP Playground. Today we're reviewing Unit 5, Heredity. So, some key points that you need to know for Unit 5 include meiosis and how it contributes to genetic variation, Mendelian genetics, including his two laws of independent assortment and segregation, non-Mendelian genetics such as different forms of dominance, linked genes, sex-linked genes, as well as being able to figure out what pattern of inheritance you see through pedigrees, and lastly, phenotypic plasticity. Alright, first up, genes and chromosomes. And this is our precursor to discussing meiosis. So a gene is a segment of DNA that codes for heredity, meaning this is going to be a gene for a trait that is transferred from a parental to a filial generation. It's transferred generationally. And so reproductive cells that transmit genes are known as gametes. So a lot of your cells are somatic or body cells, but we have gametes, and those are going to be the ones that transmit genes, the reproductive cells, the egg and the sperm. And so sexual reproduction is where two parents contribute genes to the offspring, and this is going to be genetic variation. The offspring is not going to look exactly the same like either parent. There's going to be a combination, and it's going to be a random combination, where the egg and sperm come together during fertilization to form a zygote. Now, genes are located on chromosomes, and this is condensed DNA. And when you have homologous chromosomes, there are pairs of chromosomes, and you get one of each chromosome from each parent. So humans, for instance, have 46 chromosomes, and that makes 23 pairs. And over here you can see in this karyotype 23 pairs of chromosomes. So this is pair 1 and so forth. And one of these is going to come from the mother, and one of these is going to come from the father. These are homologous chromosomes. And 22 pairs of chromosomes are autosomes, and one pair is sex chromosomes. These are the chromosomes that determine biological sex, whether it be XY or XX. And so human somatic cells, non-sex cells like we discussed, all have 46 chromosomes. And this makes them a diploid cell, otherwise represented as 2N, because they have both of the sets. They have the 23 chromosomes from the mother and the 23 chromosomes from the father, so they have all 46 chromosomes. Whereas gametes, the sex cells we discussed, the egg and the sperm, they only have 23 chromosomes. They are haploid cells. They are known as N, because each one has half the chromosomes, so when the two gametes come together, the egg and the sperm come together during fertilization, we result in offspring that's going to have 46 chromosomes. And so now we're going to be discussing meiosis, and that's the process where one chromosome of each pair is given to a gamete randomly. Meiosis is how these sex cells are formed, so then sexual reproduction can occur. So now let's look at the stages of meiosis. So now we're going to talk about the different stages of meiosis, beginning with prophase 1. But first we must talk about S phase, where replication of DNA occurs, just like it does in mitosis. So when we look at our homologous chromosomes, our one paternal, one maternal, they're going to be attached by a centromere, and during replication, each homologous chromosome is going to gain a sister chromatid. So now there's two paternal sister chromatids and two maternal sister chromatids. So now we're still looking at 46 chromosomes, but there's also sister chromatids for each chromosome. So, and meiosis is going to have two stages of division, and the result are going to be 4 genetically different gametes. And of course, it starts with a diploid cell. It starts with our 2N, that is then replicated during S phase, and then two stages of division during meiosis 1 and then meiosis 2 result in haploid cells that contain only a single chromatid. So let's begin by talking about prophase 1. Chromosomes are going to condense just like they do in the beginning of mitosis. And then homologous chromosomes are going to attach, and what's called crossing over is going to occur. So here we have our homologous, here's our maternal and paternal chromosomes, and each one has the sister chromatid from DNA replication. But what's going to happen is these internal sister chromatids are going to attach at what are called synaptic complexes, they're going to form crisscross regions called chiasmata, and what's going to happen is DNA from one chromosome is going to be cut and exchanged with the exact portion from the other chromosome. So what we see here, after crossing over has occurred, is that all 4 sister chromatids within this one chromosome pair are different, because we have our initial paternal and maternal chromatids, but we also have a crossed over portion that is genetically different from the other 2. And this is a significant contributor to the genetic variation that meiosis provides. So now let's look at the rest of the steps of meiosis. So continuing on with meiosis 1, where we talked about prophase 1 and crossing over, next up we have metaphase 1, where the homologous chromosomes are going to line up at the metaphase plate, at the center of the cell. This is very similar to metaphase within mitosis, and we're going to get independent assortment. So if you look here, here's prophase 1. We had our synaptic complexes form and crossing over, and then we're lining up at the metaphase plate. But there's independent assortment, meaning that there's going to be a random orientation on which side the maternal and paternal homologous chromosome is going to be. So here, for instance, we have 2 of the, let's call these the maternal chromosomes, and a single paternal chromosome, whereas on the other side of the cell we have lined up at the metaphase plate 2 paternal and 1 maternal. And this is a 50-50 chance for whatever orientation the homologous chromosomes are going to be. And this matters because during anaphase 1, when the homologous chromosomes separate and get pulled apart, and eventually telophase 1, where nuclei rebuild and we've got now 2 separate cells, that means that there's going to be a different amount of paternal and maternal chromosomes on each side of the cell, and this is like a random chance thing that can occur. And then during meiosis 2, which is to separate each of these chromosomes, to separate the chromatids within each of these chromosomes, again, we're beginning with a haploid cell. Prophase 2 begins with a haploid cell because we've gone from having both the maternal and paternal chromosomes in one cell, from our diploid 2N cell here, to having partial, it's not entirely either maternal or paternal, it's mixed due to independent assortment and crossing over, but we only have half of the chromosomes now. And then again, sister chromatids are going to line up at the metaphase plate in metaphase 2, they get pulled apart in anaphase 2, and lastly in telophase 2, they're going to reach opposite sides, nuclei are going to reform, we have cytokinesis, and once this happens for both of the cells generated in meiosis 1, now we're going to be ending up with 4 haploid cells that are all going to be genetically different. Now, comparing meiosis and mitosis, we only have synapsis and crossing over in meiosis, because in mitosis we're not trying to create genetic variation. Cells are replicating exactly. There's no genetic difference between cells. Additionally, we don't go through 2 phases of division. We don't have meiosis 1 and meiosis 2 because there's no focus on pulling apart homologous chromosomes. During metaphase and mitosis, sister chromatids are pulled apart and that's it. Whereas in meiosis, sister chromatids stay attached during anaphase 1 and are only pulled apart in anaphase 2. So connecting meiosis to genetic diversity, we discussed how crossing over, independent assortment, those are going to result in increased genetic variation because we're mixing up the paternal and maternal chromosome. And why this is important, each diploid cell is going to have 2 different versions, 2 alleles of the same gene. There's going to be 1 allele that comes from the mother and 1 allele that comes from the father. And so sexual reproduction causes these alleles to be mixed up. We're producing varying offspring. There's genetic diversity, and that can help survival. And we talked about crossing over. We talked about independent assortment, but also random fertilization. Each combo of egg and sperm is unique. So after meiosis has occurred, the random, like which of the egg and which of the sperm are going to fertilize to create a zygote, there are 70 trillion combinations, which is absolutely insane, and that creates so much variation within population and within the human species as a whole. And why it's key to have genetic variation, we can see, for instance, here with our peppered moths. And so these guys were living around the time of the Industrial Revolution, and they would often land and gain nutrients from trees that had pretty light bark. So the ones that were better camouflaged were the lighter peppered moths. Those were the ones that could camouflage from predators such as birds that would eat the moths. However, once the Industrial Revolution occurred and factories started pouring out smoke and ash and soot, there was all of the trees were blackened and got darker, and as a result the bark was no longer light colored. And so now the moths with the advantage, with the survival advantage with the camouflage, those were the dark colored moths. Those could survive better because they were now the camouflaged moth. And so if there was no genetic diversity, if all the moths were light colored, then when the Industrial Revolution occurred, the birds would easily be able to pick out the moths that land on the trees because they're all light colored. But because of this genetic diversity, the population was able to survive an environmental change. And we'll talk about this more in Unit 7. But for now, it's crucial to remember that meiosis helps create genetic diversity through crossing over, through independent assortment, through random fertilization, and this is beneficial because it creates variation within a population. And now we must discuss genetics. And this starts with Mendelian patterns of inheritance. So genetic information is carried by DNA and RNA. And these are going to affect the outward traits of the organism, because whatever DNA, whatever your genotype is, your genome, your DNA codes for, that's going to be the proteins that are produced and other molecules that are produced that are then going to shape your outward appearance, your phenotype. And heredity, which is the title of this unit, is the chance of offspring inheriting the traits of parents. So what is the chance that the offspring is going to inherit certain genes, certain DNA, that will then contribute to a phenotype that makes them look like a parent or not? And so Mendel made discoveries about genetic inheritance through breeding pea plants, both through the flowers and the pea pods. And so his observations include the following. There are alternative versions of the same genes, like we talked about in the last slide. There are different alleles. And so they code for the same thing, but they're slightly different DNA. And he saw this with purple and white peas. And he determined that each parent contributed one allele to the offspring. Homozygous organisms have two of the same allele for a trait, and these could be dominant alleles or recessive alleles. And how he explained what a dominant allele was, this was an allele that would be expressed over the recessive allele. If we look in this picture here, where he bred a purple pea flower and a white pea flower, then all of the offspring were purple. And so purple is the dominant allele for the pea plant color trait. And so heterozygous organisms, though, have two different alleles, do different traits for an allele. So they're not going to be entirely dominant. So for instance, let's say we're going to call color for the pea flower color, that's going to be C. So the dominant allele is going to be capital C and the recessive allele is lowercase c. So we can have homozygous dominant, homozygous recessive, and also heterozygous. That's going to be a mix. But any heterozygous organism is going to express the dominant allele because the dominant allele is going to take precedence over the recessive allele. And so we can represent these ideas within a Punnett square. And for instance, when we're looking at this purple flower here, that's going to be a homozygous dominant flower. And there's going to be one allele from each parent, one dominant allele from each parent. And now this white flower is going to be recessive. There's going to be one recessive allele. There's going to be one recessive allele here from each parent that goes into the formation of the offspring. And of course, the recessive allele can only be exhibited when the offspring is entirely homozygous recessive, because if there is a dominant allele present, then the dominant allele will be expressed. And when we do this crossing in what's known as a Punnett square, we can see that each of these offspring organisms are going to be heterozygous. They're going to have one dominant and one recessive allele. And we can see that represented here with our 4 purple flowers. Now, they don't have the same genotype, they don't have the same genome as the parental purple flower, but they do have the same phenotype. They both have an outward appearance of purple. And now Mendel also came up with some laws that explain the Punnett square, which we'll take a look at now. So now discussing Mendel's laws, we'll start with the law of segregation, which states that each offspring has a 50% chance of inheriting either allele from the parent. And the alleles segregate, they separate, meaning there's a 50% chance for the offspring to inherit this dominant allele and a 50% chance for the offspring to inherit this recessive allele. And this goes for both parents, and that's why the Punnett square works this way. Now, if the organism is heterozygous or homozygous dominant, but you're not sure, it displays the dominant phenotype, but we don't know the genotype, then you can determine what the genotype is by crossing with the recessive organism. Because depending on whether it is heterozygous or homozygous dominant, then there's going to be a different genotypes and phenotypes that come out from the offspring. And we can do that Punnett square here. So let's say we have our flower, which can either be homozygous dominant or heterozygous. And it's going to be a red flower for the dominant allele. Now the homozygous recessive organism, should be a C, homozygous recessive organism is going to be a yellow flower. So when we do our Punnett squares, if it were to be a homozygous dominant organism paired with this homozygous recessive, then all of the offspring would be heterozygous. There would be no chance of having an offspring that was not heterozygous. However, if this were to be instead a heterozygous organism that is being crossed with the homozygous recessive organism, then there are going to be heterozygous offspring, but there are also homozygous recessive offspring. And based on this offspring, you can now tell what genotype the parental organism has. Now we've been looking at one trait, and when you cross two heterozygous organisms and you're looking at one trait, that's known as a monohybrid cross. That's what's shown up here on the right in this Punnett square. And there's a ratio rule that there's always going to be a 1-to-1 genotype. So there's always going to be 25% or around 25% homozygous dominant, 50% heterozygous, and 25% homozygous recessive. Those are the general percentages of offspring for two heterozygous individuals. And of course the phenotype is going to be a 3-to-1 ratio. There's going to be about 75% that are dominant because that's going to be the dominant phenotype, but the genotypes are split up between homozygous dominant and heterozygous. Now down here we can also look at a dihybrid cross where we're looking at multiple characteristics. And we're looking at both the yellowness or greenness of the pea, but also the roundness or how shriveled the pea is. So we are crossing two individuals that are heterozygous for both of these traits. And what allows a dihybrid cross to work is the law of independent assortment. Each pair of alleles will segregate separately, which just means that alleles for different genes aren't linked. And that allows all of these gamete combinations to occur. We can have the alleles for both traits to be dominant, an allele for one trait to be dominant and the other recessive, the other dominant and the first one recessive, or both recessive traits. And so when we do this dihybrid cross, we get a 9-3-3-1 phenotypic ratio where 9 organisms are going to exhibit the dominant appearance, and then 3 are going to exhibit dominance for one, recessive for the other, 3 are going to exhibit dominance for one, recessive for the other, and then 1 is going to exhibit recessive for both traits. Last thing we need to discuss is calculating probability. And this goes for asking, are we needing to add to find the probability, or do we multiply to find the probability? And you add when there are independent events. If you're trying to find the probability of this or that, then you can add the probabilities together. But if you're trying to find the probability of a combined event, usually that's signified with the word and, then you're going to be multiplying. So the example here is if we're going to cross an individual with this genotype, meaning homozygous dominant for A and heterozygous for B, with this genotype, which is heterozygous for both, what is the probability of getting an organism that is going to be homozygous dominant for both traits? And so the way we can do this simply, instead of having to draw a dihybrid cross, is we can find the probability of getting a homozygous dominant for the A allele, or the A trait, and then homozygous dominant for the B trait. And because we're trying to find the probability of this combined event of an individual that has both homozygous dominant for the A and homozygous dominant for the B, then we can multiply those probabilities in order to get our answer. So we can set up our Punnett squares for this right over here. So let's do the A's first. We have our homozygous dominant and heterozygous. So that's going to create homozygous dominant. That's going to be homozygous dominant. And this is going to be heterozygous, which means there's a 1/2 chance, a 50% chance of getting homozygous dominant for the A's. Now we can take a look at the B's and go through the same process. These are both heterozygous individuals. So this is going to be homozygous dominant, then heterozygous, heterozygous, and homozygous recessive. There is a 25% or 1/4 chance of getting homozygous dominant for the B's. So then if we want to find the probability, the total probability of homozygous dominant for both traits, then we need to multiply the probabilities. So 1/2 times 1/4 gets us a probability of 1/8. Now it's time for some math. And we calculate chi-square to compare expected and observed values in the data to determine if there's a significant difference between the expected and the observed. And this can be applied to genetics when we discuss whether or not the offspring that resulted from a combination of organisms reflected the projected Punnett square statistics for the inheritance of those traits. And the goal here is to reject the null hypothesis. And the null hypothesis states that there is no significant difference between the two. And so if the null hypothesis is rejected, that means there is a significant difference between the expected and observed values. So the chi-square equation is right over here. This is our chi-square. We see our sigma or sum symbol right over here. And for every single point of data, we need to subtract observed minus expected, square that value, divide it by expected, and add up all of these values in order to get our chi-square value. And then we have to compare it against a value on the critical values chart. So the level of significance that we'll be using for this course is 0.05, and that means that you're like 95% sure. There's 95% surety of whatever you are calculating. There's 5% potential room for error. And your degrees of freedom are going to be the number of classes minus 1, meaning that if you're looking at 4 different options, then you would have 3 degrees of freedom, and the comparison value you would be using is 7.815. And then you could compare your chi-square value to 7.815. And if the chi-square value were to be smaller than this critical value, that means that there is no significant difference between the two. And we will be looking at an example here, so it's okay if you're not entirely clear on the concept yet. So the question is as following. A poker dealing machine is supposed to deal cards at random, as is from an infinite deck. In the test, you counted 1,600 cards and observed the following. Spades 404 cards, hearts 420 cards, diamonds 400 cards, and clubs 376 cards. Could it be that the suits are equally likely, or are there discrepancies too much to be random? So is there a significant difference between what the expected value would be, or do the expected and observed values match enough for there not to be a significant enough difference? And so first we have to begin by, obviously we know what all our observed values are, and we have to talk about what the expected values are. If we have 4 suits at random out of 1,600 cards, then there is an equal chance of pulling each suit of cards. So there should be 400 spades, 400 hearts, 400 diamonds, and 400 clubs. And as we can see that for some individual points of data, for instance, spades are fairly close. Diamonds are exactly the expected value, but hearts and clubs are not as close to the expected value. So now we can calculate chi-squared to tell if there's a significant statistical difference. So, for instance, for the first point of data, we have our observed, which is going to be 404. Let me put that down here so there's enough space for future calculations. 404 minus the expected, which is 400, squared over the expected, which is 400. And when you do the math, you get 404 minus 400 is 4. 4 squared is 16. 16 over 400 is 0.04. And if you'd like to pause the video now and try the rest of the problem yourself, you can do so. I'm not going to write out the math for every single step because it's always just following this formula of observed minus expected squared over expected. And in the case where you do get a negative number, for instance here where it's 376 minus 400 squared over 400, well, that's part of the reason why the squared is there, because then that gets rid of the negative. If you square a negative number, you now have a positive number. But if you do this for every single, for all of the suits of cards, then you'll get 1 for, you'll get 1 for the hearts, you'll get 0 for the diamonds because there's no difference, and you will get 1.44 for the clubs. And the last step is observing this sum notation symbol. That means we need to add up all of these values. And adding up all of these values gets you an answer of 2.48. Now we have to find the right value on our chi-square chart in order to make this comparison. So we're using our column of 0.05 level of significance, and now how many degrees of freedom are we looking at? Well, we have 4 different options that the poker dealing machine could have randomly chosen from. So that means we have to take 1 away from 4. That means there are going to be 3 degrees of freedom. There are 4 options minus 1 makes 3 degrees of freedom. And so we look at our value of 7.815 and we need to compare it to 2.48. Obviously, 7.815 is larger than 2.48. We can add a 0 there as a third decimal point. And so what this means for us is that there is no significant statistical difference between the expected and observed values. The poker dealing machine is actually dealing cards at random, and the difference in the observed values are not significant enough to suggest that the poker machine is broken or potentially cheating. However, Mendelian genetics are not the only patterns of inheritance seen in nature. They're sort of the simplest versions of inheritance. So when we looked at Mendelian genetics, we were discussing complete dominance, where the homozygous dominant and heterozygous are completely indistinguishable. The dominant allele is always going to be taking control there. It has complete dominance over the recessive allele. However, these aren't the only forms of dominance. There's also incomplete dominance, in which heterozygous organisms demonstrate a phenotypic mix between dominant and recessive alleles. So there's an in-between value. For instance, with these flowers here, homozygous dominant red and homozygous recessive white create a heterozygous pink. There's a mix between the homozygous and between the dominant and the recessive in creating the heterozygous phenotype. There can also be codominance, where 2 alleles are dominant and affect the phenotype in 2 different but equal ways. So both alleles are technically dominant. Here, with our red and our white flower, we see codominance displayed here, where there's going to be some petals that are red and some petals that are white in the offspring. Both alleles are dominant, one is not covering up the other. Additionally, we have multiple alleles. Which are genes which exist in more than 2 allele forms. So more than having just one dominant and one recessive allele, there are multiple allele forms. And the most common is human blood type. There's an A-type blood, there's B-type blood, and there's an I. And so the different combinations here that we can see with genotype, if you have an AA or an A lowercase I, with the I being sort of the recessive allele here, then we get A-type blood. Same thing with if you have BB or B lowercase I, then you've got B-type blood. If you have two of the recessive alleles, then you've got O-type blood. And if you have an A and a B, then you're now experiencing codominance. You have AB-type blood. Both of these alleles are dominant. Lastly is polygenic inheritance, where two or more genes have an additive effect on a single character and phenotype. So a single trait, for instance, if we're taking these flowers again, like the color of the flower is not affected by a single gene. Multiple genes have an effect on the phenotype of the flower. And one example of this in humans is skin color. There's not one gene that codes for skin color. It's an additive effect of multiple genes. Moving on with non-Mendelian patterns of inheritance, we got to talk about linked and sex-linked genes. So obviously, as we've discussed, genes have specific locations on chromosomes. So on the chromosome, there's going to be a specific site called the locus, which is where that gene is going to take place. And linked genes are on the same chromosome and tend to be inherited together during cell division, because they are on the same chromosome. And really, the only time that they become separated is when crossing over occurs in prophase 1 of meiosis. And so when offspring inherit the phenotype of parents, they're considered the parental types. They've inherited the sister chromatid that is entirely the parental type. And if they inherit the chromatid that is the recombined one due to crossing over in meiosis, then they're known as recombinants. And genes that are further apart from each other have a higher chance of recombining. They have a higher chance of separating through crossing over in meiosis 1. So if we look over here, if we have meiosis with no crossover, then we have 4 parental chromatids at the end because no genetic mixing has been done. But if there is a crossover, then what's going to result is 2 parental chromatids, but also 2 that are recombinants, 2 that have mixed DNA because of crossing over. And the further apart genes are, and we'll talk about mapping genes and determining how far apart they are on a chromosome on the next slide, but the further apart they are, the higher the chance they have of recombining. And there are also sex-linked genes, which is a gene that is located on a sex chromosome, so an X or a Y chromosome. And generally, we're going to be looking at X-linked traits. So sex-linked genes are either X-linked or Y-linked, and X traits can be recessive or dominant in daughters, but always represented in sons. And what this means is that in a daughter, you have a chance to be, let's say this is a recessive trait. So you could have one dominant X-linked gene, let's say for the letter H, and one recessive allele, and that's going to mean that you are not expressing this trait. You are a carrier of the trait because you have the recessive allele, but you're not expressing this trait. However, men don't really have that option. Biological males do not have that option. If there's a trait on the X chromosome, then it is the trait that is going to be expressed because there is no other X chromosome that could potentially mask that trait. And fathers pass their X-linked genes to their daughters, but not their sons, because sons will get the paternal Y chromosome. So if we look over here, there's this male who is dominant for this X-linked trait, and there's the Y chromosome, and then the female who is dominant for the X-linked trait in one allele and also recessive in the other, so a heterozygous individual for the X-linked trait. And so offspring can either be a daughter that is going to be homozygous dominant, a daughter that is heterozygous, a son that is dominant for the trait, or a son that is recessive for the trait. So if this is actually a recessive trait, then one male offspring will be affected because unlike the daughter, which can mask up that trait through the dominant allele, the daughter or the son is not able to do so. Let's practice some linked genes. So gene 1 has 2 possible alleles, a dominant A and a recessive A. Gene 2 also has 2 possible alleles, a dominant B and a recessive B. An individual of genotype dominant for both, heterozygous dominant or homozygous dominant for both, is crossed with an individual who is homozygous recessive for both, yielding dihybrid offspring that's going to have a heterozygous genotype. and then one of the dihybrid individuals is test crossed with a homozygous recessive genotype. So question number 1 are going to be the percentage of recombinant and parental organisms that result from this crossing. So parental organisms are going to have the same genotype as either of the parents. So that's going to be this guy who has these guys who are homozygous recessive and these guys who are heterozygous, while the recombinants are going to be the ones that do not have the same genotype as the parents. And we can calculate the percentages for the parental by adding up all of the organisms with the parental genotype, and that'll be 800, dividing the total, which is 1,000, and then multiplying by 100 to get the percent, and that leaves 80%. And that makes the recombinant percentage 20%. And now, so when asking whether these genes are linked or not, you could do the Punnett squares, look at the Mendelian patterns of inheritance. That would be your expected data. Then look at your observed data, which we have in this table here. Do a chi-square analysis to determine whether there's a statistically significant difference between the expected and observed data, and arrive from there to the conclusion that these genes are in fact linked. But you can also look at the percentages, and they're pretty bleak here. We can see pretty clear that 80% of organisms are having the parental genotype, and only 20% don't. There is very little genetic variation within the offspring, and this is going to suggest that these genes are on the same chromosome and that they were not separated during crossing over. Now, the number of map units is how far apart on the chromosome they are, and the number of map units is equivalent to the percentage of recombination frequency, which is just the percentage of individuals of offspring that are recombinants. So we determined that 20% of the offspring are recombinants, and so we have a 20% recombination frequency, and these genes are 20 map units apart. That is how far apart they are on the chromosome. Now we also have to take a look at pedigrees, which is a diagram that shows the relationship between parents and offspring across 2 or more generations. And so we have our generations here, first, second, third generation. We have individuals in each generation. It represents biological sex. We have females that are circles and squares that are males, and we can also look at certain traits where the shaded individuals express the traits. And this can help predict the genome of future offspring. Now, the question asks, what kind of trait is this? And so, when we're looking at this, pedigree, we can see that over here in 2.4 and 2.5, these individuals had children. They both had the trait. All of their children had the trait. Now, individuals 2.1 and 2.2, one of them has the trait and one of them doesn't. However, none of the children have the trait. And that suggests to me that this is going to be a recessive trait. Because if one of the parents were to have the trait and they would have children, then some of the children would have to have the trait, because it would be, if it were to be a dominantly expressed trait. However, if this were to be a recessive trait, then it would be masked up by another individual who doesn't have the trait, and that could result to something like we see here in 3.1, 3.2, and 3.3, where these organisms don't have the trait even though one of their parents does. Last up is phenotypic plasticity and non-nuclear inheritance. So genetic traits and phenotype can be affected not only by the genetics, but also by the environment, and phenotypic plasticity represents this. Individuals of a population that have the same genotype can have different phenotypes in different environments. We can see this with skin tone in humans. When you're tanning in the sun, even if you have an identical twin, but if they don't tan, then you're going to have a different outward appearance, even if your genetic information is exactly the same. You have the same exact genotype. Flower colors can change based on the pH of the soil. The seasonal fur color in Arctic animals, such as this Arctic fox, change to allow it to better camouflage in different environments during different seasons of the year, where it's better for the fur to be white during the winter when it's snowing and gray during the spring when there isn't as much snow on the ground. Inheritance also doesn't have to come from the nucleus. DNA can exist outside of the nucleus, such as in mitochondria and chloroplasts, which contain genetic information. And so mitochondrial DNA is not passed down through meiosis. It's passed down only through the egg, because the sperm does not have viable mitochondrial DNA. So what happens is through the mitochondrial DNA, which is sort of known as maternal DNA, because you can trace a maternal line using mitochondrial DNA as it is only passed down through the mother. So there you have it. That's Unit 5. We'll see you in Unit 6 to talk about gene expression and regulation.